Hi everyone, my name is Tyler Daniel and I'm a second year student at Columbia University studying Medical Humanities, and today I will be sharing my presentation on genetics. So with regard to what we will talk about today, we'll discuss what genetics are, where the genes are located in the human body, what genes are, what are alleles, and what are probability charts, also commonly known as Punnett squares. So what is genetics exactly? Genetics, simply put, is the study of how genes and traits are passed down from one generation to the next. So it may be helpful to think about genetics as a building block for a house. So genes are a blueprint to instruct our bodily processes on how to function. Therefore, the genes that we carry impact our health, appearance, and personality. So, for more of this discussion on the genes and where they're located, we're going to start off on a larger level and then we're going to narrow our way down. So, we're going to start off with the nucleus, then we're going to go to the chromosomes, then the DNA, and a little something I like to call the alphabet that deals with base pairing. So, the nucleus is probably one of the most evident cells in the human body, one of the most commonly known, I'm sure. Given its internal complexity, the nucleus is a membrane enclosed organelle that is found in most eukaryotic cells. And these are the cells that are found inside us, humans, along with animals, plants, fungi, and protists. As one of as the largest organelle in the cell, the nucleus contains most of the cell's genetic information, so it's really important when we're discussing genetics. Before we move on to the chromosomes, I actually wanted to address the difference between eukaryotes and prokaryotes. And the reason why this slide succeeds the slide on the nucleus is because one of the main differences between the two cell types is whether it has a nucleus or does not have a nucleus. In the case of eukaryotes, which are cells for humans and animals, for example, there is a nucleus that is present, and there are also membrane-bound organelles that are present. For prokaryotes, there is no nucleus present, and there are no membrane-bound organelles present either. And a good example of this are bacteria cells. So one way to think about the differentiation between membrane-bound organelles or no membrane-bound organelles is kind of thinking about like a cubby or a compartment system, whether you have some sort of like organizational system in your room or on a desk, where you can put all pencils in one area or all markers in one container, all crayons in one container. Instead of having those compartments, those membrane-bound organelles that are pretty much encompassed in certain sections, prokaryotes, they don't have those. So you can kind of think of like laying all your crayons down on your desk and all your pencils and all your colored pencils, and so having them like distributed across your desk. I feel like that's a good way to think about the difference between eukaryotes and prokaryotes when it comes to their membrane-bound organelles versus their non-membrane-bound organelles. The next part of this discussion leads us to the chromosomes. So, chromosomes are a set of genetic material that the nucleus holds. There are typically 46 chromosomes total, consisting of 23 pairs. And the reason why I say there are typically 46 chromosomes is because sometimes there are genetic variations that either deviate from that 46 number, so that means it could either be higher or lower. Uh, with that, there are different functions that the body goes through with regard to a different number of chromosomes from the 46. In order to ensure that genetic material is present, chromosomes go through a process of division called mitosis. And this is when the genetic information from parent cells, where they begin, are divided until each cell receives half of the original information. And this half of the original information makes this really familiar X shape, as we can see on the slide. And chromosomes are formed through the condensing and coiling, so this makes the DNA itself smaller and more compact, and that change that allows us to see this X shape. And it's really cool because this X shape is something that we're really familiar with, something that, although it doesn't look too much like this cartoon drawing, but it does have a faint resemblance when you look at chromosomes under a microscope of some sort and enlarging the image. And so it's quite cool that DNA, being so very long, can be condensed into the shape that we can study further. So before I move on to the slide on DNA, would like to use this slide as maybe a question and answer between the students and the teacher, maybe pause for some brief discussion or reflection, see if there was information that maybe stuck out to you as new, something that was interesting, maybe this was a great review for you and how did you feel about it. There is a fun fact on this page, and it's that chromosomes help determine the sex of an individual at birth. So feel free to ponder using this slide before moving on to DNA. Right, so with DNA is a very, very short way of saying deoxyribonucleic acid, which is much more longer and more complicated to say. But DNA is basically the genetic material within cells. This is the information that was passed on from your parents and determines your characteristics. And so the discovery of DNA as genetic material was very important in the findings of the field of molecular biology and really just understanding how information is passed on from parents to offspring. And DNA also has a very iconic, if you will, shape with the twisted ladder that resembles a helix-like shape. And we'll get more into, as we go into the next couple of slides, more into how that shape actually forms and what the process is going on in order to get this helix that we have. Again, feel free to use this page to, as discussion, ponder a little bit, share some interesting information that you may also have. So I think this is a great way to reflect. There's a lot of information, a lot of new topics that are being shared, so this might be a wonderful way to pause and digest as well. Yet again, there is a fun fact on this page, and it takes approximately 8 hours for one of your cells to completely copy its DNA. So if you kind of think about that, it takes about 7 to 8 hours is like probably the recommended amount of sleep. So in the amount of time that you sleep, DNA can completely copy itself, which is quite impressive because DNA is quite long. So now we're at the page called the alphabet, and it is not necessarily referring to the 26 letters that we are all familiar with. Rather, the term alphabet in this context, I'm using it to describe the nitrogen bases that compose DNA's double helix that was mentioned earlier. Nitrogen bases are part of a larger structure called nucleic acids. These are also known as macromolecules and are essential instructions for your DNA to carry out to form unique codes that then make proteins in your body that provide us with the traits that we have. So it may be helpful to think of this code similar to how a video game animates characters and gives them actions. And so the base pairing that occurs is giving your DNA the instructions it needs to carry out certain actions. In order to have this code, if you will, work, nitrogen bases need to bond with its complementary pair. The nitrogen bases that we have that are present include adenine, cytosine, guanine, and thymine inside of DNA. They're all denoted with their letters respectively, adenine as A, cytosine as C, guanine as G, and thymine as T. So adenine always pairs with thymine in DNA, while cytosine always pairs with guanine. I like to remember this with the phrase apples in the tree, cars in the garage. So that means A is pairing with T, C is pairing with G. With the complementary pairs in place, the strands of genetic information combine, creating a twisted ladder shape that we know as the double helix. And given how long DNA is, I think the next slide actually has a fun fact about its exact length. But given how long DNA is, it and how many base pairs it has, it is inevitable that there might be mistakes that occur. And so there are both biological processes as well as new medical technologies and scientific discoveries that are being done, or research rather, that is being done to try to alleviate any detrimental or harmful effects that might come from misunderstood base pairing, so the DNA can still function in its intended way to carry out certain bodily processes and allow and provide that blueprint for our body to function. Again, another page for questions, reflection, discussion, just to digest some information. But the fun fact on this page is that DNA contains approximately 3 billion base pairs. So you can imagine that's a lot of base pairs. DNA is quite long again, and so with having that many base pairs, again, it is inevitable that mistakes may occur, but there are ways that those mistakes can be fixed. So, what exactly are genes? And to put it short, genes are a small segment of DNA that carries information that is then transformed into a chain of protein, which is the alphabet, the nucleotide bases that we were discussing before. These genes are fundamental to heredity, which is the passing of traits from parents to their offspring. So this really can explain why you have features similar to your parents. If you've ever gone out and someone's like, wow, you really look like your mom, or wow, you really look like your dad. The passing on of genes and these instructions and how they encode information is very responsible for someone's characteristics. What are alleles? So the term alleles are as used to describe the alternative form of a gene. So alleles are located on the arms of chromosome. So there are the P arm and a Q arm, and that's just given the names given the length of the chromosomes themselves. And an individual is able to inherit 2 alleles for each gene. So that is one from each parent. Alleles can either be homozygous or heterozygous, as we'll get and discuss more in the next couple slides. And we'll also look at the probability charts or Punnett squares and how that relates to either being homozygous or heterozygous in some cases. So being homozygous and heterozygous, if both alleles are identical, the individual is homozygous for this gene. And so that means if they are both the same from both parents, so let's say someone gets one allele from one parent and another allele from one parent and they're the same, that makes that person homozygous. If both alleles are different, that means one allele from one parent and one allele from another parent, but they are different, they're not the same, that individual is heterozygous for that gene. So now we're going to look into probability charts that are also known as Punnett squares. And these provide a summary of possible combinations of alleles from the mother and the father. These tables are typically used to examine the genotypic outcome for offspring through single or multiple traits. So to provide some more context on what the genotypic ratio might look like or what exactly that means, it really focuses on the more microscopic level of someone's genes. And so, for example, we're using R's in this case on the slide. And so those might help depict what a genotype may be. They're something that we can't really see. They're more on a DNA level. That is something that the human eye won't be able to discern. However, the phenotypes of offspring can be assumed whether alleles are present, which are homozygous dominant, homozygous recessive, or heterozygous. And so those phenotypes are what are expressed, what people can see. For example, someone has brown hair, blonde hair, like a reddish strawberry color hair color. Someone has brown eyes, blue eyes, hazel colored eyes. Those are your phenotypes. Those are what we can see and what are expressed based off of the genotype that you have that we can't see, but we can maybe infer what your genotype might be based off of the traits that you're expressing. So in this case, with the probability chart or Punnett square that we have a parent. We have two parents who are heterozygous for a certain trait. And the reason why we know that they're heterozygous is because they have one big R and one little r. And so typically when you do a probability chart, you use letters of some sort. I really enjoy using letters that look different in their uppercase versus lowercase writing. And so I felt R was a good example for this. Some letters such as like S or C or even X, they're really hard to discern because they don't have a different style depending on being uppercase or lowercase. It's just really a size difference, and so that can make it complicated when you're carrying out a Punnett square and trying to figure one out. And so, with regard to the parents being heterozygous, we can then use this Punnett square to try and figure out the possible genotypes of the offspring, and from there, pretty much infer what their phenotypes could be. And so, the genotype, so if we look at the upper left quadrant, we see that both parents would donate big R like alleles for that gene, right? So that person would have two big R's and be homozygous because they're the same, but they'll be homozygous dominant because that uppercase letter is indicative of a dominant gene. And so they would be homozygous dominant for that gene. In the upper right quadrant and the lower left quadrant, the one parent is donating an uppercase R, and then one is donating a lowercase r, and then vice versa. One of the other parents is an uppercase R, one of the other parents is a lowercase r. And so in that case, the offspring is heterozygous, meaning that they have different alleles for a particular gene. And so as a result, they are heterozygous. And last but certainly not least, in the lower right quadrant, that offspring has two small r's, so two lowercase r's for their alleles, because both parents donated two small r alleles. And so in this case, the offspring is homozygous, but they're not homozygous dominant, they're homozygous recessive. And so since they have two little r's, so little letters or lowercase letters are typically indicative of recessive genes. And so in most cases, the dominant gene will override or be a bit more stronger than the recessive gene. However, if a trait is only expressed by recessive genes and someone has two little r's, for example, or let's say two little like B's or two little A's, they would actually express that trait. So this pretty much segues into a conversation about phenotypes. So being dominant or having a big allele pretty much overrides, again, it's much more powerful, if you will, if you put this into like a tier list, much more powerful than the little r. So the big r's, right, even though there's a 25% chance of the offspring having big R, and then a 50% chance of the offspring having big R, little r, there's a 75% chance that they show the dominant allele. So that's their phenotype, right? They show that dominant allele because all three of them have the big R, and the big R is going to be stronger and be expressed over the little r. Now there's a 25% chance that the genotype is little r, and there's also a 25% chance that the phenotype that's expressed is the recessive phenotype for this offspring. So I know that was a lot of information, and Punnett squares can be a little daunting at first, but I think there are, with a lot of more practice, and you'll see as we go through the rest of the slideshow, I feel like you'll be able to get the hang of it. And so I'm going to use this slide as maybe a method to pause and reflect, think about what was just shared, and get ready for a few activities that are yet to come in this presentation. And so the fun fact on this slide is that DNA is used in genetic engineering to achieve specific traits and characteristics. There are ongoing conversations with regard to the ethics of certain practices and how this might affect society on a larger level more than just on the basis of science. And so there's a lot more research and discussion that's going on that it's really important with regard to DNA and doing anything to really change it and how it may function. So let's see genes in action. So we're going to learn how we can use Punnett squares to see the genotypes and how that correlates to the probability of children expressing a specific phenotype from the traits passed on to their parents. So I know I spoke a little bit about this with the examples of the heterozygous parents that had a big R and little r allele and going through that Punnett square, but I would love for students and particularly the teacher, whoever is present, to pick some volunteers if anyone is interested. And using these traits, we have widow's peak versus no widow's peak, dimples versus no dimples, cleft chin versus no cleft chin. It'd also be really cool if a student has one of these characteristics and want to use their parents as examples, and feel free to choose your own letters for this. Again, my recommendation is using letters that you can really tell apart versus uppercase and lowercase. It's much makes doing the Punnett squares much easier. And yeah, just have a really good time with this and putting what we've learned into practice. Now, this is another interactive activity. It's a little different because this is a virtual presentation. I'm not in person to really help facilitate this. However, your teacher should have access to this slideshow as well. But this activity allows you to either choose to make a pet fish or a pet bird, and it's really talking about how certain traits might be present if you decide to do dominant or recessive traits. So feel free again to make more Punnett squares. Depending on which animal you choose, you'll get a different set of characteristics. For example, the fish, you'll get scales, while the bird, you'll get feathers. And so you can decide, your teacher can peruse through, decide which which traits are present, and then as a class, create Punnett squares and see, even if it's at random, see what the possible phenotype could be and choose to make your pet that way. Feel free to also grab some blank pieces of paper if you want to do this in groups, and draw out your animals as well as the prompts come up on the screen. I think it's a really nice way to generate more discussion and get some more practice with these Punnett squares. Right, so here are the resources used in this slide. As always, I think it's very important to, with anything that you do, obviously, citing where information comes from. And this allows people who are listening to a presentation or reading a paper of any sort to reference where you got your information from and maybe even learn more from those sources as well. Right, and this is just another recap, a little bit about me. I'm happy to connect with students that may be interested in learning more about genetics in general, or even if you want to learn more about college, high school, have any questions about that. I would love to share any of my experiences or be a resource to you all. So your teacher should also have access to my email. So if anyone's interested, feel free to reach out. I would look forward to hearing from you guys. And a feedback survey. Feel free to share how this presentation was for you. It is, again, it's a little bit different doing it in a virtual setting, but hopefully it was very interactive and you were able to learn a lot or even maybe refresh on some topics. Maybe this was a really nice review. Feel free to leave some feedback on things that can be improved, things that you really liked. It really helps as we continue to do presentations in the future. Thank you so much.